It's nice to be back. I graduated from University of Wisconsin-Madison like almost 5 years ago. So it's always nice to come back and share with everyone what has been going on with my own group at MD Anderson. Oh, and also like special acknowledge to Zheng, who from Daifeng Wang's group. She's very kind enough to switch this very valuable session with me so I can catch flight back home tonight. So very special thanks to her. Okay, so I'm going to share with you my recent work on the FFPE stuff. So you probably have heard of these words, FFPE. So it's a short for formalin fixed paraffin embedded. So if this is not the most popular, but must be one of the most popular way to archive tissue and especially store it in the room temperature for a long time. So the FFPE procedure start with like in the operation room, doctor take a tissue or tumor from patient and put into formalin and then covered by paraffin so that it can be stored in a room temperature for years or even decades. So showing here are some FFPE samples. You can see like in the center, there's some tissue or some tumor and then covered by the paraffin. And sometimes you can scrape it off and it will curls up so it can put in the tube. You can do all kinds of experiments in the tube directly. So on the other side of easy storage, FFPE is also known for its damage on the tissue. So for example, for RNA-seq molecule, so when when they are put into formalin, there can be some formaldehyde cross-linking that lead to those RNA molecule being chopped up. And also in the paraffin, there are some organic solutions. They will also continue to chop up the RNA molecule. So over the years, the humid or temperature will also continue to degrade those RNA molecules. So that's why, like experimental speaking, like it's very hard to extract the intact RNA molecule and have an unbiased portfolio on that. So that's why, like, we propose a new technology called FFPE-CUTAC that can profile the RNA-PO2 on DNA. So it's kind of like a DNA assay. So the RNA-CUTAC is kind of like a special version of cut-around-cut attack, but specially designed for FFPE sample. So the good advantage is that it can give you a very short fragment that is around like 120 base pairs. In other words, even though the DNA will also be chopped up by FFPE procedure, because FFPE-CUTAC procedure will zoom in and only give you a very small fragment. So it will be less impacted by the FFPE damage and degradation. And also because we are mirroring the RNA-PO2 directly on DNA, so you can directly know the activity of a gene translation, a gene transcription. So this is like a direct measurement of gene activity. This is not using RNA accumulated in the cell as a approximate of the gene activity. This is like a direct measurement of gene transcription activity. In other words, this technology will be free from all kinds of RNA drawback, for example, RNA variable half-life transcript and RNA instability in the cell. So with this technology, we're curious, how can we use such a powerful tool to study human cancer? So we profile 7 human cancer. We have their tumor and adjacent normal tissue for comparison. So always, the first step is to see whether we get like high quality data. What is the data quality? So first, like we have shown here like 7 like tumor type, and as expected, like using this technology, we can easily separate out not only tumor versus normal, we can also separate each cell type. And also showing on your right-hand side, this technology is also free from all kinds of biases like sequence bias, coverage biases. And when we really look at those regions of scientific interest, you can see like on the genome track, background is also minimal, sharing all those cut-around-cut attack new technology advantage. And also for those regions that do have hypertranscription, you can see like, for example, highlighting here is a HER2 gene region is the RNA-PO2 signal is highly enriched for breast and colon tissue. So good. For those regions of interest, it does shows the signal in a relatively reliable way. And speaking of the hypertranscription, we also notice there's another group of regions that shows consistent hypertranscription. Those regions are histone genes. So regardless whether it is in the mouse tissue or in the human tissue, regardless what kind of cancer type we are profiling, we always observe hypertranscription, like high enrichment RNP2 in those histone genes. Showing here, red are tumor, blue are normal tissue. So regardless whether it is like brain tumor, a different type of brain tumor for mouse, or different like human cancers, for tumor tissue, it always has high histone signal. High RNP2 signal on histone genes. So that motivates us to see how can we explain such observation. Because histone genes, as we know, like it's encode histones. So for cell replication, not only we need to replicate the DNA, we also need to generate the histone so that DNA can wrap around the histone to form nucleosome. Because like nascent DNA is quite fragile and dangerous. And if the cell cannot wrap around, form a nucleosome, the cell will die. It will be the same for both cancer cell and normal cell. So that's why like for cancer cell, it also need to, because if cancer cell want to proliferate in a very aggressive, fast way, it not only need to replicate the DNA, it also need to generate the histone in a very aggressive and fast way. So that's why, even though like for us human, we only have like 64 histone genes, it's kind of encode all 5 histone subunits that are needed during the replication of the S phase. So that's why we kind of hypothesis that the histone production can be the rate limiting for the S phase for the like cell replication progress. So our hypothesis is that the hypertranscription that we observe not only in mouse or human or across different human cancer, those hypertranscription in cancer cell is to produce enough histones to keep up with the requirement so that the DNA can wrap around the histone form nucleosome so that cancer cell can proliferate much faster than normal cell. So faster cancer proliferation need fast generation of histone genes, need hypertranscription in histone genes. Okay, so biologically it seems to make sense, but why hasn't been studied for so many years? Why, if histone genes is important, why it is understudied for so many years? This is because like for the very traditional RNA-seq assay, the RNA molecule signal capturing needs the RNA, the poly-A tail capturing. But the replication coupled histone, because it needs to generate in such a fast manner, so it has its own machinery. It does not add poly-A tail towards the end of RNA molecule. That's why like those histone genes product in RNA format cannot be captured by the traditional RNA-seq. So that's why it's kind of overlooked over the years. Okay. So using FFPE-CUTAC, this new technology, we can profile the histone gene signal. How can we use histone genes to predict clinical outcome? Because we kind of like hypothesis the histone gene signal is the indicator of how aggressive the cancer can grow, right? So how can we use it to predict the clinical outcome? So the system we choose is meningioma because this is kind of like the most common brain tumor. But the special characteristic of this tumor is that most patients with meningioma are benign. Most tumor are benign. However, for those patients who come back with recurrent tumor, those tumors tend to be very aggressive, tend to be highly like malignant. However, the histologic feature looks almost the same as the benign tumor. So that's why we're thinking like with histone genes, can we provide some assortment information, some better prediction of whether this patient will reoccur or not. Okay, so the data is that we use our new technology, FFPE-CUTAC, to profile 30 meningioma patient. The drawback is that those patients are relatively new, so there's no recurrent information yet. So very minimal clinical data associated with this sample. But the good news is that over the literature, over so many years study of meningioma, we can collect almost like 1,300 public RNA-seq data. And those data sets can be profiled from frozen RNA-seq or small proportions of profile use FFPE sample. But the good thing is that they all come with very rich clinical outcome, especially the recurrent time and status. So if we can borrow the information from the literature, from the RNA-seq data, then we can see whether histone genes can predict certain clinical outcome. Okay, so after getting the data, again, first is quality check. So we want to see like whether we can separate out different cell type. So showing here, highlighting here using the flag symbol, they represent the meningioma sample. So if we use the whole genome and bind them by 500 base pairs, you can see like we can already have separation across different tumor type. If we zoom in and only focus on those dysregulatory regions that ENCODE provide, you can see the separation is even better. And again, meningioma form its own like cluster, meaning that they are separate, they're different from all kinds of our other cell type, tumor or normal sample. However, if we zoom in again, only focusing on the 64 histone genes, you can see that cell type information are gone, but there is a clear pattern separating like tumor versus normal, meaning that some information seems to be shared across different type of tumor, especially within those 64 histone genes. So that motivates us to continue to see whether histone genes, for example, there are some tumors that tend to be located closer to normal, are they more towards like benign tumor? For those that are very far away from those normal sample, are they tend to be more aggressive tumor? Okay, but again, FFPE sample itself does not have clinical information. We need to borrow it from RNA-seq data. So how to integrate two modality data? So integration is not like stranger to computational biologists. We use canonical correlation analysis, CCA. It's kind of like developed for single cell data, but we found it's also working for integrating two bulk cell bulk modality data. So we use CCA to integrate the FFPE-CUTAC and RSG. And you can see here, like if a sample starting with a symbol like C or A, those represent patient number. It's ending with CUTAC, meaning this is FFPE-CUTAC. It's like RNA potent profiling on FFPE sample. If it ending with frozen, meaning it is RNA-seq profiling on those frozen sample. So you can see as long as it's from the same patient, it tends to be clustered very closely with each other, almost on the overlap pattern. Yeah, which means using CCA, we can nicely integrate FFPE-CUTAC with RNA-seq. So now we put two modality data on the same shared embedding. We can use those shared embedding to borrow the clinical information from the RNA-seq data. Okay, so how can we use histone genes to predict the borrowed clinical information? So first is that we have 64 histone genes. I just do a very simple summary of all the signal on the 64 histone genes and get one signal. So showing here is a density distribution of all those like almost like 36 samples. So in total, there are 36 score. So you can see like most of the patients will have score around this area. So if I want to set a threshold to call those are predicted to be like aggressive tumor, those are predicted to be benign tumor, how should I set the threshold? So that motivated me to go back to the nature characteristic of this cancer, this meningioma cancer, is that most cancer are benign, which means if I want to set a threshold, it's better to set at the right-hand side of the peak, right? So that most of the patients are still benign. So I just randomly put a threshold on the right-hand side of the peak and call those patients with high histone signal. We predict them to be aggressive tumor, and the rest are like benign tumor, and see how the clinical information tells us. So again, like with this very arbitrary, very data-driven thresholding strategy, 5 patients with high histone signal are given the aggressive label. So they are highlighting here using the red dot. So the blue dots or blue triangle are predict to be benign sample. So dots are FFPE-CUTAC, triangle are RSG data. So we borrowed the 20 nearest neighbor from the RSG data and use their clinical information to see whether we can separate out aggressive tumor and benign tumor in terms of the survival or the recurrence information. So to our surprise, like it's separated pretty well. So all those patients who are given aggressive label, their RNA neighborhood tell us like those patients recur very fast than the benign predicted to be benign patient. And also, I just look up in the Wikipedia, and it says like around 10 to 15 patients tend to be like a very aggressive meningioma tumor. So this range definitely aligns well with our very data-driven thresholding result. It's like 5 out of 36 samples are predicted to be aggressive. You probably are curious, like, because it seems to be so arbitrary to set a threshold here. I can set a threshold a little bit to the right or a little to the left, right? So to us, the thresholding is more like a large data training problem. However, here we only have 30 patients. So setting a threshold is not our top priority. Making sure that histone genes can give us a good order, that's our top priority. Is that whether histone gene signal is a reliable indicator. So for those patients with very high histone gene signal, are they tend to be more aggressive versus those with low histone signal, they tend to be more benign? This is more important question to us than giving a very fixed threshold to call aggressive or benign. So that's why I explore all kinds of threshold, going for a more stringent to more aggressive, to a more like relaxing thresholding strategy. So you can see if I set the threshold here, fewer patients will be called aggressive. Oh. So that's why like here, maybe it's too small, but only 3 patients are deemed aggressive, but you can see the survival curve very distinct. And going forward, when I set a very relaxing threshold, like more patients are deemed aggressive, so you can see the separation gradually converge, which means like the order actually matches with the clinical outcome. So higher histone signal tend to be associated with more recurrent, like fast recurrent clinical outcome. And we definitely have some negative control and positive control because we're curious whether other genes also have such a prediction power. So we check with ribosomal protein genes, no separation, chromosome M, because there's this Warburg theory saying that maybe low percentage mitochondria tend to predict like high aggressive cancer, but still no separation power. And chromosome 22q is kind of known. Chromosome arm loss for meningioma patient is like almost 70% of patients with a meningioma patient will have chromosome 22 loss. So with that, that's our positive control. Definitely this can have separation power. Okay, so we have only tested on meningioma sample, but it seems like histone genes is kind of promising in predicting the clinical outcome because it's kind of like the rate limiting bottleneck for cancer cell to proliferate. Going forward, we're going to establish an experimental and computational component for this FFPE ecosystem. Yep, definitely. If you have some FFPE sample and know someone who have FFPE sample who can benefit from this ecosystem, feel free to reach out. With that, I want to thank all the lab members in my postdoc group and also all the funding because I just started at MD Anderson, so I'm hiring at all levels. So spread the word. Thank you.
Is there a relationship between genomic instability in the samples and some pattern that you're finding?
Genomic instability, what do you mean?
Rearrangements and other types of
Yeah. So we also have another project starting the aneuploidy and see how it can lead to. So because we already know his aneuploidy can cause cancer or like aggressive cancer. So our hypothesis is that whether histone genes is hypertranscription lead to aneuploidy. Yeah.